Hi everyone, you're welcome again to class. It's always an interesting time meeting you. Thank you for all the commendations after your practical exams. Today, we'll be looking at some selected questions. We have called out some questions on different topics in biology SSCE just to make sure that you prepare adequately as you go into the examination hall. So in our normal way of doing it, we are going to be making sure that we display these questions to you periodically and we'll walk through the answers together. So I would advise that you get out your reading materials, your writing materials, your pen, your biro, and maybe a bottle of water to just sit as we travel this long distance together. It's going to be a lengthy one, but it's going to be worth the while. If you're a new person today, thank you for joining us. Thank you for coming by. We promise that at the end of this, you're going to have so much value and you're going to be better prepared for your examinations. If you've not subscribed, kindly help to click on the subscribe button, like, share, and comment on this video so it can reach as many persons as possible. We are really grateful for your supports all along, and then we promise that we are going to continue delivering value to you as we travel together. So if you are ready, I am ready. Let's get started. Question number 1 says, what is classification of living things? Now, if you are a biology learner, you will not have any issue defining classification of living things because it's one of the first topics we look at in biology. The next says, name the scientists that developed the binomial system of classification. So you're going to see that as well. The next is, state 3 reasons why it is important to classify living organisms. And the next one says, list 7 major groups into which taxonomies classify living organisms in their hierarchy. So you have to follow the order. The next question says, state 7 characteristics of living things and explain each of them briefly. So you have to see that and see how you can answer those questions for yourself. Let's look at the answers very quickly. So looking at the answers, you'd see that classification of living things is the sorting or grouping or arrangement of living things into groups based on similar or shared features or characteristics. So you are grouping them based on the things they have in common, and that will give you your 2 marks. So sorting of living things or grouping would give you 1 mark, and then similar characteristics will give you another 1 mark, making it your 2 marks. Name the scientists that developed the binomial system of classification. You have to spell correctly. So this is how to spell it correctly. Carolus Linnaeus or Carl Linnaeus in short. Just make sure that you don't miss a word. If you miss a word, you'll be penalized for doing that. Now the next question says, state 3 reasons why it is important to classify living organisms. So the important reasons for classifying organisms are, number 1, for easy identification of the similar living things, and then next one, to show similar relationships between organisms, to trace geographical distribution of organisms, and then you also use it for education, for research, and then for easy reference. So note those things down as we travel together. These are questions that have appeared over the years in SSCE exams, and we are expecting some of them to come up as you prepare for the paper. The 7 major groups into which taxonomies classify living organisms in their hierarchy are, so we have the kingdom, we have the phylum, we have the class, we have the order, family, genus, and species. So the kingdom is the broadest, okay, it has the least characteristics, and then you have the species. The species is the commonest, okay. Organisms of the same species share the highest number of similar characteristics. So this has the most characteristics and this guy has the least characteristics. Okay, so moving on to the next question. Let's see the next question. The next question was 7 characteristics of living things and explain them briefly. So let's quickly see a few of them here. The first one is movements. We already know movements is a change in position, otherwise known as locomotion, when the whole body is moving, and then plants cannot move from their whole body, so they only move parts of their bodies. Respiration is the breaking down of glucose to release energy needed for metabolism. So you can pause and write that down. If you don't know what that is, you should know. And then nutrition is the process of obtaining food. This can be autotrophic as found in plants and heterotrophic as found in living animals basically. Irritability is the response to stimulus by organisms. These stimulus could be internal stimulus or they could be what? External stimulus. If it is internal stimulus, that way we talk about the autonomic nervous system and the likes. Okay, so let's quickly move to the next question. The next question says, in a tabular form, state 4 differences between plants and animals. That's also very primary. You should be able to do that. The next one says, name one organism that has both plant and animal features. Take note of this question. And then the next one says, state 4 plant-like features and 2 animal-like features of the organism you mentioned here. So when you mention the organism here, you have to mention 4. This is supposed to be 4 animal-like. That's a mistake. This is 4 animal-like features and 2 plant-like. Okay, and 2 plant-like features of this organism. So that's what we're going to look at there. So let's quickly see what we have as the answers to that. The remaining characteristics were growth, okay, and then excretion, removal of waste, metabolic waste from the body, and you also had reproduction, giving birth to individuals that look like you basically. So let's come here to see the differences between plants and animals. We talked about the fact that when you're marking these differences, any difference at all in biology, it is marked at the center. So you must get the 2 points on both sides correct for you to score your mark. So here we said locomotion is present here, and then here they cannot move their whole body, they only move parts of their body. Okay, that's for plants now, and this is animals. And then animals obviously cannot make their food, but plants can make their food by photosynthesis. Animals do not have a cell wall, but plants contain a cellulose cell wall. And then basically animals have your respiratory and excretory organs, but plants don't have specific ones. Take note of that. Now the organism that has both plant and animal characteristics is the euglena. You know this already. Okay, the love-shaped euglena. So what are the plant-like features of euglena? We started with the animal-like features. You want to write this down, they're very important. So the presence of the flagellum for movement is animal-like. It also has a gullet for taking in food, that's the second one. It has a pellicle for flexibility, that's the third one. And then it has the contractile vacuole for excretion. So these are the 4 animal-like characteristics of euglena. And the 2 plant-like characteristics are that it has the chloroplast for photosynthesis, and it has the pyrenoid for storing of what? Of starch. This is very, very important. It comes almost every year, so please take note of that. Okay, so moving on to the next question. The next question says, state 5 major phyla in the animal kingdom and give one example of each. So phyla is the plural form of phylum. Okay, the phylum we listed earlier after kingdom, that's what we are saying here. So we're going to see 5 major ones, but I had to bring this gap because I want to mention all of them for you in case you are asked. Now the next one says, mention 2 levels of organization in living organisms, giving one example of each. Okay, so we're going to see that as well. 2 levels of organizations. Here we have from the cells to the tissues to the organs, and then to the what? To the system. Remember I say you can pause the video at every point, attempt to do this by yourself, and then before you look at our answers. Now the next one says, what is complexity of organization in living organisms? And then state 3 advantages of this complexity. So you have to look at that again. So let's see that and see what we have as our answers. Looking at this now, you have the phyla. So we have the porifera. This is where porifera or the coelenterata, that's the first one here. An example there is the sponges. You have the platyhelminths. You have the example in the platyhelminths is your tapeworm, your planaria, or your liver fluid. And the next one is the nematoda, annelida, mollusca, arthropoda, echinodermata, and your chordata. You want to write this down. This has come very recently in your exam, so please take note of this. Know these worms, know the examples, and then get a few of them. Take note of phyla arthropoda. Arthropoda has a very good interest because it contains 4 basic classes. That's where you have the cockroach, the insecta, the myriapoda, the arachnids, and the crustaceans. So please take note of this particular phyla, and of course this is where we belong. Take note of the chordata. All mammals are in the phyla chordata. The next one is, I told you this now, levels of organization of living things. So you have the cell. This is number 1, this is number 2, this is number 3, and then this is number 4. You also have the organism now. So the organ system makes up the organism. Okay, so but the cells, you have the amoeba, the red blood cell, the nerve cell. I mentioned this guard cell. Guard cell is an example of a plant cell, and you also have the root hair cell. So the root hair cell is another example of the plant cell. For the tissues, you have the xylem, the phlegm, the blood, the bone, and the muscle. And the organ, you have the eye, the nose, the stem, the roots, and the leaves. And then for the system, you have the excretory system, reproductive system, digestive system. In the plants, you have the roots and the shoot system. So this is for the what? This is for the plants. So take note of that. Now the next one says, what is complexity of organization? Basically, this means that organisms, this is the arrangement of living things in levels where the simple structures combine to form more complex structures. So you see, amoeba, all these ones that are unicellular, they have a very simple structure, but as you're going down, they become what? They become complex. So that's basically the idea of complexity of what? Of organization. Alright, so I think we've answered all the questions there. Okay, 3 advantages of complexity. We are going to see it in the next slide, but let's go to the next question now. The next question says, complete the table by mentioning the class of vertebrates in their evolutionary trend and one of the example of each. So basically, the vertebrates here are members of the chordata phylum. Okay, the last phylum we mentioned on the second page was what we were asked to list the examples there. So we have the 5 classes. We're going to see that in a bit. And then we came to cells. What is a cell? And then state 7 basic organelles found in a typical plant cell and state their functions. You want to be writing these things down. If you're not writing these things down, you're very wrong. You're not doing yourself any good. So get a pen and a paper and settle down. Put these information down. They will really help you in your examination. So let's quickly look at the answers to see some of those things. So looking at this here now, you have, okay, these were the advantages of the complexity. We talked about the fact that there is division of labor, there is greater efficiency. So because organisms are now complex, there is efficiency, and each organ can do its function. The eye can have the function of seeing, and then the ear can have the function of hearing and balance. This is not found in lower organisms. And then there is better survival adaptation. The organisms can survive better because they have this specialization. And then the classes we talked about earlier, you see the Pisces. Example of the Pisces, you have the tilapia, the tuna, the catfish. You have the amphibians. The amphibians examples are the frogs and the toads, the reptilia, the lizard and the snake, the apes, the dove, the pigeon, and then you have the mammalia. Now, all the spellings must be correct to score. Sincerely, if you fail to write these spellings correctly, you will be penalized. Your marks will be taken away from you. So please take note of this, is very important. Okay? And then, what is the cell? The cell, of course, is the basic structural and functional unit of life. Okay? It's the basic word structural. Let me just put that down very quickly. And functional what? Units of life. So please take note of that. It's very, very basic. You can define a cell. Now the cell organelles we talked about, you have the cell wall. Remember they say in a typical plant cell. So you have the cell wall. What's the function of the cell wall? To protect the cell and give it a rigid shape. You have the ribosome. Some of you are hearing these things for the first time. You have to just write these things down and take note of them in case they come in your exams. The ribosomes are the site for protein synthesis. What do you mean by protein synthesis? The manufacture of protein. This is where protein is manufactured. And then you have the almighty mitochondria, otherwise known as the powerhouse of the cell. This is the site for respiration. And after respiration, ATP is produced, and that's what helps you to walk around. Okay, that's what gives the energy you need for all your daily activities. I hope you are getting some value already. Alright, so we are not done with that, but let's move to the next slide. We are going to continue. Now the next one says, explain briefly the cell theory. The cell theory is one concept. They asked these questions about 2 years ago in the NECO biology. So please you need to pay attention to the cell theory, know it. And the next question is also very, very interesting. We now ask you to name the stage 5 scientists who contributed to the development of the cell theory, their contribution, dates, and we have this column here. We have their name, we have their contribution, and you have the date of discovery. You want to write down this table. Every information on this table is very, very vital. So let's quickly see a flash of what we've got for you. Now, this is what we've got for you. The other organelles, of course, that we look at on the other page, you have the nucleus containing the genetic material, the vacuole storing excess food and water, chloroplast for photosynthesis, and then endoplasmic ER actually means endoplasmic reticulum, okay, and then that helps in the transport of materials within the cell. The cell theory states that number 1, all the cell is the basic structural and functional unit of life. Number 2, all plants are made up of cells. Number 3, all animals are made up of cells. And number 4, cells come from previously existing cells. So those are the 4 theories that make up the cell theory. In case you're asked, write that down and get your full marks. Now, these are the dates you need to pay attention to. Okay, so these slides or this particular document has been very, very detailed. We took our time to make sure that if they bring in any question on cells, you should be able to squash it and get all your marks for allotted for the question. Now, Robert Hooke was the first person. He observed 18 slides of cork tray under a microscope, and he named its tiny compartments as cells. Basically, he was a monk actually, and then so that was in 1665. Matthias Clay then as the first person that proposed that all plants were made up of cells, and he did that in 1838. Theodor Schwann was the next person. He proposed that all animals are made up of cells, and he did that 1 year after Theodor Schwann, Matthias Klein rather, and that was in 1839. And then this guy now, very great guy, Rodolfo, he came up with the statement that cells come from pre-existing cells, meaning cells today will become 2 cells tomorrow and they'll give birth to their new cells, and that was in 1855. So please take note of these dates, they would come in your exams. Alright, so we're moving on to the next one now, the next question. The next question says, state 7 specialized cells and their functions. See, I have to bring in specialized cells because learners over time or students have an issue identifying specialized cells. Specialized cells are cells that have a unique structure, okay? They have a unique structure that is different from the conventional cells. And I hope you know, I just come to my mind now, I should mention to you that there are 3 basic organelles that you have in a plant cell that you don't have in an animal cell. So I hope you know them. You can pause the video and list them down. So that is your chloroplast, your what again? Your cell wall, and then that was your large vacuole actually. Okay, so these 3 organelles are always found in a what? In a plant cell but not in an animal cell. So coming back to specialized cells, we're going to look at a few of them and see their functions. And then the next question is, identify or define briefly the process of diffusion, osmosis, and active transport. We're a bit fast because we have a lot of things to cover and we want to see how we can work with the time we have. So you can pause the video periodically, write these things down, and then follow through with your questions. Alright, let's see the answers to that set of questions. So basically, specialized cells, you have the red blood cells in animals, white blood cells in animals, the nerve cell or the neuron there's an E here, okay, a neuron, the muscle cell, the sperm cell, and these 2 are in plants, okay? So you have the root hair cell in plants and you also have the gut cells in plants. What do they do? Their functions are all listed here. This is in your SS1 biology or your 10 biology, so you should be able to put this down very well. Red blood cells transport oxygen, white blood cells defend the body. It has 2 ways of defending the body, either by producing what is called phagocytes or by producing antibodies or by producing antibodies. The lymphocytes produce the antibodies, and the neuron or the nerve cells help to transmit impulses around the body. The muscle cells contract to produce movement. Sperm cells fertilize the egg cell. The root hair cell absorb water and mineral salts, and the gut cells control the opening and the closing of the stomata. Alright, so let's move on to the next one. Diffusion basically is the movement of particles from a region of higher concentration to a region of lower concentration. Okay, that's diffusion. When particles move from when they are in high concentration to where they are in low concentration. In osmosis, this basically means the movement of water molecules. So that's what you should know. Osmo represents what? Water. So osmosis is the movement of water molecules from a region of high concentration again to a region of what? Lower concentration through a selectively permeable membrane. So this is now the difference. There has to be a selectively permeable membrane. So maybe I am trying to do a little sketch here. This is my region where I have my high concentration of water molecules, and here I have very low concentration, but there has to be this membrane dividing it. So when they go from this high region to this low region, this membrane is now the selectively permeable membrane. That membrane was not there in diffusion. Okay, so take note of that. And then active transport, on the other hand, now means the movement of substances from the region of lower concentration to the region of higher concentration using energy. Okay, so now when substances move from where they are high, from this place where you have about 4 of them to where you have about 10 on this side, now they are moving against concentration gradient. So write that down. This happens against concentration gradient, and concentration gradient simply means the difference in concentration between the two regions. Okay, so moving on to the next question. If you don't understand some of the things I am saying here, you have an option of putting it down in the comment section. Let's try to see how you can interact and make you understand better, or you write them down and check them out on Google yourself so that you can follow up and be very fast with what we are doing. Now the next question says, explain what happens when a red blood cell is put in a hypotonic solution. Okay, and the next one here says, state 3 differences between turgidity and hemolysis. These are terms you should know as a learner preparing for your NECO examination. The next one says, what are autotrophs, and give us 2 examples of your autotrophs. So let's quickly see the answers very quickly. So here you have that when a red blood cell is put in a hypotonic solution, what happens? Water enters the cell by osmosis. So a hypotonic solution is a dilute solution. So write that down. A hypotonic solution is a what? Dilute solution. It's the reverse of hypertonic. Hypertonic solution is a concentrated solution. So we are asked what happens when it is put in a dilute solution. So when this red blood cell is put, because the solution is dilute, water will have to enter into the cell, okay, by osmosis, and then the surrounding has a higher water concentration than the cell content. I was trying to explain here. So the cell will swell up and may eventually burst, okay, may eventually what? Burst. And then that's what happens when a red blood cell is put. When a plant cell is put in a hypotonic solution, it doesn't burst, and that's simply because it contains the cell wall. That's what we are capturing in the next question. So turgidity happens in plants, whereas hemolysis happens in what? In animals. Okay, so take note of that. So I wrote it here: occurs in plants, occurs in animals. So can absorb water and becomes firm. This one, excess water will make it to burst. The presence of rigid cell wall prevents bursting, and the absence of cell wall allows the cell to do what? To rupture. So that's the difference between turgidity and what? Hemolysis. Go back and check other words like flaccidity and crenation. Okay, they will help you as you prepare. The next one says, what are autotrophs, and give 2 examples. So basically we have here that autotrophs are organisms that can manufacture their own food from simple inorganic substances. They either use light, if it is light, it's photosynthesis, or they use chemical substances, which is thermosynthesis. And examples of autotrophs are your grasses, your green plants, and then you have your algae. Another example of what? Your autotrophs. So take note of that. Moving on to the next question, we are making progress, and I hope you are getting some value. I hope you are writing some things down already. Now the next one says, explain carnivorous nutrition with 2 examples. Carnivorous nutrition in plant. That looks very funny, right? How can a plant be a carnivore? So we're going to see that in a short while. And then here I had to bring this again. What is chemosynthesis? Because I needed you to understand what we mean by that. Now, we want you to talk about this. Define the term photosynthesis, and then The next one, list 4 external factors and 2 internal factors that affect the rate of photosynthesis. So let's see the answers very quickly. So looking at that, you see that a carnivorous plant or carnivorous nutrition in plants is a mode of nutrition in which certain green plants trap, kill and digest insects or small animals to obtain nutrients, especially nitrogen, while still carry out photosynthesis. So you want to pause this video and write this down. They are plants. Normally plants make their food from photosynthesis, but these carnivorous plants, they still have a deficiency in their nitrogen level, so they need to take in insects. Okay, they have different mechanisms of taking in insects just to supplement for that extra nitrogen. Common examples are your Venus flytrap, your pitcher plants, your bladderwort, and your sundew. These are all examples of what? Carnivorous plants. Now there are other groups of plants called parasitic plants. Parasitic plants are the ones that don't even have to make their food by photosynthesis. They are different from carnivorous plants, and a good example of that is your what? Is your doda or your mistletoe, what you were given in your WAEC specimen earlier in your series of exams. All right, so the next one says, what is chemosynthesis? Chemosynthesis is a biological process by which organisms like your bacteria and your archaea, they convert carbon molecules. So they convert carbon molecules like carbon dioxide and methane into organic matter. This is not like photosynthesis, because photosynthesis relies on sunlight, but chemosynthesis relies on the oxidation of inorganic chemical compounds such as hydrogen sulfide or methane. So the conversion of these carbon molecules into organic matter is what is called chemosynthesis, and it's carried out by bacteria and archaea. Okay, now what is photosynthesis? Photosynthesis is the process by which, of course, green plants make their food, which is glucose, from carbon dioxide and water in the presence of sunlight and chlorophyll, releasing oxygen as a byproduct. Okay, so basically that is what you have as photosynthesis. Now the external factors that affect the rate of photosynthesis are light intensity, carbon dioxide concentration, temperature, and water availability, whereas the internal factors are chlorophyll content of the leaf and then the leaf structure or the number of chloroplasts. So take note of that as you prepare for your exams. So let's move to the next question. It says, outline the process involved in the light and dark stages of photosynthesis. So pay attention to these kind of questions because they come periodically and students get to struggle with answering these kind of questions. Now the next one says, an individual ate a hot and large meal of cassava and a salty groundnut soup. Okay, the individual was sweating profusely and did not drink water after the meal. Name 2 end products of the meal after digestion. And then the next one says, state 3 advantages that the individual will derive from the meal. So we ate a meal of cassava with salty groundnut soup, and then he was sweating and did not drink water. So that's an applied question. So pay attention to what? Application questions. They are going to be asking you some form of application questions. You can see they ask you to plot a graph in your WAEC. That has not come for a very long while. Okay, so the exam bodies are beginning to tilt towards application questions, so pay attention to answering these kind of questions. That's why I had to bring that there. So let's see the answers very quickly. So quickly, the light stage or the light dependent reaction of photosynthesis involve the following process. Number 1, chlorophyll absorbing the sunlight. The light will split water into your hydroxyl ion and your hydrogen ion, and that is what is called photolysis of water. Oxygen is then released as a byproduct. You can just write that down, and then your hydrogen is produced and energy is trapped and stored as ATP. In the dark reaction stage, the carbon dioxide is absorbed by the plants and then it combines with hydrogen that was released in the light reaction stage. Then glucose is now formed from what? Simple sugar, or glucose or simple sugar is formed. Now for the hot meal and the hot cassava meal and the groundnut soup, here the end products of the meal digestion obviously will be glucose because there was a cassava, so glucose would be one of the end products after the digestion, and amino acid because there was a groundnut. So that's basically what we have there. 3 advantages that the individual will derive from the meal. Of course, the glucose will give him energy for his body activities. The protein will give him and will repair and help in the growth of his tissues. And then of course you will still get some mineral nutrients needed for his normal bodily activities. Okay, so that's is that for that question. Let's move on to the next question very quickly. The next question says differentiate between macro and micro elements. So pay attention to these questions. I was intentional in bringing this question because I know these are areas that the NECO exam body would want to go to test your ability to understand these concepts. And then again, I asked here, I grouped this into, and most of these questions came from your past papers. If you are reviewing your past papers very well, you will be able to recognize some of these questions. So here I grouped some elements into your macro elements and micro elements, and you're asked to tick the ones that are macro and the ones that are micro as required by what? By plants. The next one says briefly explain the mode of feeding in tapeworms. So you want to be writing these things down. Don't be tired of writing. Be writing them down as you review them for your exam. All right, so let's see the answers very quickly. Now you see that macro elements are those required by organisms in large amounts. Macro means large. Okay, and examples are your nitrogen, your potassium, your phosphorus, while your micro elements are the ones required by the organisms in minute or small amounts, and examples are your iron, your zinc, and your copper. So you have this now as a table to really bring this into categorization. So let's see the ones I ticked for the macro. So the macro, you have your hydrogen, you have your magnesium, you have your phosphorus, and you have, of course, these three ones, your CHO, nitrogen, your carbon, and your oxygen. And then the micro are your what's this one? Zinc, your copper, your manganese, and your what? And your boron. So the next one says describe briefly the mode of feeding in tapeworms. Basically, tapeworm is parasitic. It is found in the guts of man and pig, okay, and it has this hook and suckers. Please write these things down. It has hook and suckers for easy attachment to the host, and it has a flattened ribbon-like, providing a large surface area for easy absorption. As a matter of fact, it absorbs food with all the parts of its body, and then it lacks a digestive tract. Of course, it doesn't need a digestive tract because it's already taking in absorbed food. So what does it need a digestive tract for? Nothing. And then of course it has no need for eyes, so it doesn't have an eye as well. So that is how to describe that to get your answers for your questions. All right, so moving on to the next question, let's quickly see how we can be a bit fast as we still have a lot of things to review. The next one says describe saprophytic nutrition in rhizopus. And then here I have this very interesting table telling us that we have the holozoic, parasitic, saprophytic, symbiotic, and autotrophic nutrition. I have some organisms here where you have to group them into all of this. So start doing that by yourself before we reveal our answers. Okay, and then finally here also we have that we have to outline the chemical test, how to test a tuber, test for starch, the tuber of yam, and then how to test for glucose in an orange fruit. Okay, so you see these questions are picked across board. So please go ahead to see how you can write these things down and take note of this. All right, so let's review the answers to see. I hope you get some of them correctly. Now, rhizopus exhibits saprophytic nutrition. So we just talked about parasitic nutrition in tapeworm. Now we're talking about saprophytic nutrition in what? In rhizopus. And the saprophyte basically feeds on decaying material, okay, dead and decomposing matter. It grows on dead and decaying matter, secretes enzyme onto the food, and digests the food externally. So the type of digestion you have in fungi, in all your fungi, mold, penicillium, and all of that is what? External digestion, okay, meaning that they secrete the enzyme externally on the substrate, and then they absorb the nutrients into their bodies, okay, and then absorbs it using the what? The hyphae. So they have this hyphae, this mass of mycelium. That is what you have. They use that to absorb the nutrients into their body. Now the table has this holozoic. Holozoic means that you are taking in substance, okay, you are taking in something. So like in the humans, we take in our food via our mouth. The Venus fly trap also takes in, it has that hole where it takes in the, what is it called now? The insect. Okay, so you have man and Venus flytrap here. Parasitic, you have tapeworm, Venus flytrap. Saprophytic, you have rhizopus, mushroom, and housefly. Of course, we know what saprophytic is. I just talked about it. Symbiotic, let me talk about this. Symbiotic has to do with a mutual relationship, basically, where when one organism benefits, the other one benefits. So why do we have lichen as an example of symbiotic? Lichen actually is a combination of two things. It has algae and fungi. Okay, so what does the algae do? The algae makes the food for the relationship, okay, because it has the green part. Remember what we talked about earlier on as an autotroph. And then the fungi provides the structure for attachment of the algae. So we see two of them now, they have the role they play. The algae is bringing the food, and this guy is bringing the attachments, or the place it's going to stay. Okay, so that's why it's symbiotic. And then autotrophic, of course, you have Spirogyra, water leaf, and elephant grass. These all have chloroplasts, and they are green in color. They can make their food. Outline the chemical steps to test a tuber of yam for starch, and then glucose in an orange fruit. So testing a tuber of yam for starch, basically you cut the yam tuber and expose the fresh surface, add drops of iodine solution, cut the surface and observe the color change. Of course, a blue-black color will be positive and will show the presence of starch. If it is negative, it's going to be reddish-brown. It will retain the color of the iodine, which is reddish-brown. And then for testing for a glucose in a fruit juice, basically, this is a Benedict test. And in Benedict test, you have to heat the solution. So it's going to be substance plus Benedict solution plus your heat. Either you use Benedict solution or you use Felin A and Felin B, it will give you the same answer. And then you observe the color change for a while. If you have a brick red, there are a series of changes, but the end product is going to be a brick red precipitate, and that's going to tell you that it is positive for that particular test. So thank you very much for your support. I hope you have subscribed. Please, if you've not subscribed, go ahead and click on the subscription button. Help us to push this video to as many persons as possible, so you all pass your exams together and come out in flying colors. Thank you for doing that. As we move to the next question. All right, so the next question says, what is a balanced diet? Okay, very interesting. Either you call it a balanced diet, or you call it what? An adequate diet. And the next one now says, state 6 classes of food and at least one importance in human nutrition. So you have the classes of food here, and we are going to see the importance very quickly. Take note of these questions. I wish I can overemphasize the importance of those questions in your exams. Now the next one says, what is a deficiency disease? If there's any word you've not seen before, just note it down, write it down, and go find the answer. That's how to be a smart learner. Now complete the table by naming 5 nutrients. Okay, 5 nutrient deficiency diseases and at least one remedy for each of them. So this is a deficiency disease and the remedy. So let's quickly look at the answers as we move on. So looking at that now, you see that a balanced diet is what? A balanced diet is any diet that contains all the essential nutrients in their correct proportions required for good health and proper growth. Okay, so all these food nutrients have to be in a balanced diet in their correct proportions. Okay, so the classes of food here, you have carbohydrates, and what does it do? It gives energy. Proteins, excuse me. And what does it do? Growth and repair of worn out tissues. Fat and oil stores energy and provides insulation. Vitamins regulates the body processes and prevents deficiency diseases. Mineral salts aids the body functions and builds strong bones and teeth. And then finally water, which serves as a medium for metabolic reactions and transportation. So please take note of that. So what is a deficiency disease? A deficiency disease basically is a disease caused by the lack of one or more essential nutrients in the diet over a long period of time, basically. And then so it means that a particular nutrient is missing in your diet over a long time, and then that will lead to a deficiency disease. So I have a table containing a few of them on this slide. We'll show you the other ones on as we go to the other one. But you have night blindness. To correct night blindness, so the remedy is to take food that is rich in vitamin A. You also have your rickets. Take foods that are rich in vitamin D and calcium especially. You can also use the early morning sunlight. And then scurvy, take foods that are rich in vitamin C, like your lemons and your oranges. Okay, they would help you with correcting that. Let's see the next question as we move on. The next question is, what is dentition? Okay, what is dentition? Explain briefly the generalized structure of a tooth in animals. So you're going to talk about the tooth and see what they look like. And then define enzymes. We have defined enzymes, and we have state 4 characteristics of enzymes. You're going to see that very quickly as well. So let's see our answers to these questions too. Now those questions now would have, okay, the remaining deficiency diseases were kwashiorkor and goiter. So write that down as well. Now dentition is the number and arrangement on type of, is the number, arrangement and type of teeth present in the mouth of an animal. So number, arrangement and type. Okay, and then explain briefly the generalized structure of a tooth. So these are the different structures you should have in a tooth. You should have the neck, you should have the crown, you should have the root, you have the enamel, you have the pulp, you have the cement. Okay, note their functions. You also have the dentine. Okay, so please pay attention to this and know the functions of each of these. We'll not waste time there. How do you define an enzyme? They are biological catalysts that are produced by living cells, and they speed up the rate of chemical reactions without being used up in the reaction. So take note of that. The enzymes are not used up. Characteristics: number 1, they are proteinous in nature. Next, they are specific in their action. The next one, they are not used up. We talked about that in the definition. And then the enzymes are temperature and pH specific, meaning they work at a particular temperature and pH. Okay, example, your protease enzymes or the enzyme in the stomach would be acidic. They work at an acidic pH, and then the ones in the small intestine will work at an alkaline pH. Moving on to the next question. Before we come here, let's see the next questions. Okay, so the next question says, list 2 digestive enzymes produced in the duodenum and in the stomach. So you have the duodenum and the stomach. We're going to see the specific enzymes that we have in these regions of the body. And then here you have this table: thiolin, pepsin, lipase, trypsin, and erepsin. You are supposed to talk about their source, talk about the substrate they act on, and the products you have at the end of the day. So we're going to see that as well. And then finally, we have now moved on to respiration. So you are supposed to define respiration. Most students have a problem defining respiration, but we're going to look at that very quickly. So let's see the answers as we quickly move on. So we see here that so we see here that the enzymes in the duodenum are your trypsin and your lipase, and then in the stomach you have your pepsin and your renin. On this table you now have the thiolin. Thiolin, the source is the salivary glands. That's where it is produced, and then the substrate is starch, the product is maltose. Remember, the end product of this is glucose, but it's not in the mouth, it's in the small intestine. That's where you have that done. So please don't make that mistake. Now the next one is pepsin. The source is the stomach, the substrate is protein, breaking it down to peptones or polypeptides. And then you now have your lipase, substrate, fat and oil, breaking it down to fatty acid and glycerol. These are the things they will ask you in your exam. They won't ask you anything other than this. So please pay attention to these things. We have painstakingly put down these resources to make sure that you are properly guided as you go into the examination hall. So trypsin, the same thing. It is sent by the produced in the trypsin, in the pancreas rather, but it released into the duodenum. The duodenum is the first part of the walls of the small intestine. You have other ones like jejunum, and you have the ileum where absorption takes place. So trypsin, here you have your protein and your peptones as a substrate, breaking it down into what? Peptides. And finally you have your erepsin also. In the erepsin is produced by the intestinal glands, breaking down your peptides that came from your stomach into your final product, which is your amino acid. Remember, amino acid is the end product of digestion of protein. The end product of digestion of starch is glucose, and the end product of digestion of fat and oil is fatty acid and glycerol. So please pay attention to that. Moving on to the next question. The next question says, explain the term oxygen depth, and then mention 2 activities that can result in oxygen depth. Okay, and then you also have here, name the 2 types of respiration, and then write a balanced chemical equation to summarize each of them. So basically we're going to see that in a very short while. Then the table now we now have a table of inspiration and expiration. Breathing in is otherwise known as inspiration. So this is breathing in, and then this is breathing out, expiration. Either you call it expiration or exhalation. And then this one, you call it inspiration or inhalation. Any of them can fly. So please take notes, pay attention to that. So let's quickly see the answers to that as we move on. All right, so having this now, oxygen depth is the extra amount of oxygen required after a vigorous exercise to oxidize the lactic acid produced during anaerobic respiration. So the activities that can lead to oxygen depth are your sprinting and your heavy weight lifting. Okay, when you are sprinting, when you are running, the rate at which the body is producing oxygen is less than the rate at which the body is using up the energy. So the body will have to temporarily switch from aerobic to anaerobic respiration. This is what we refer to as oxygen depth. And types of respiration. So you see that aerobic respiration occurs in the presence of oxygen, and anaerobic respiration occurs in the absence of oxygen. Okay, so for aerobic respiration, you have C6H12 O6 plus 6O2 to give you your 6CO2 plus 6H2O plus your ATP basically. Okay, for anaerobic respiration, the products will now be ethanol and carbon dioxide for the one we call fermentation, and then the other one, the fermentation occurs in microorganisms like yeast and bacteria, but your anaerobic respiration in your muscles will lead to what is called the lactic acid. The lactic acid we had that led to this oxygen depth in the first place. Okay, so moving on to the next question, inspiration and expiration. These are the processes that occur in inspiration. The diaphragm contracts and flattens in inspiration. The rib cage moves upward and outward. Thoracic cavity volume increases, the pressure decreases, and because of that, air moves into the lungs. Of course, when there is an increase in volume, there will be a decrease in pressure, but the direct steps occur during expiration. The diaphragm becomes dome shaped, meaning that they become curved. Okay, that's a dome shaped diaphragm. Okay, it becomes curved. The rib moves downwards and inwards. The thoracic cavity volume decreases, the pressure in the lungs increases, and then because of the pressure, increased pressure, air is what forced out of the lungs. So that's basically what you should have there. All right, so I hope you are writing these things down, and I know you are getting some value already. All right, the next one is, what is excretion? What is excretion? Mention or explain the 3 stages in urine formation by the kidneys. Okay, 3 stages in urine formation by the kidneys. The next one is, complete the table below, and the table has the organisms, it has the structure for excretion, and it has the names of the waste products that they excrete. So you're going to see that in a very short while. So looking at that very quickly, you're going to have this now. Excretion is the removal of metabolic waste products from the body. Okay, metabolic waste product and toxic substances from the body. And the 3 stages involved in urine formation are ultrafiltration, check this out in your textbooks, selective reabsorption, and tubular secretion. Either you call it tubular secretion or you call it your hormonal secretion. And the hormone we are talking about in this case is your antidiuretic hormone. Okay, so antidiuretic hormone, ADH. So you need to read about that. So but what happens in ultrafiltration? Here, water, salt, glucose, and urea are filtered into the Bowens capsule. These are low molecular weight substances, LMW, low molecular weight substances. The high molecular weight substances like your protein and your blood cells cannot pass down. Okay, they can't pass down because they have a larger size than this, except you have a defective kidney. So selective reabsorption, when these substances pass down into the Bowens capsule, some of them are useful. For instance, water is useful, all of glucose is useful, and some amount of salt as well. So when these substances enter back from the proximal convoluted tubule back into the bloodstream, that is what we call selective reabsorption. Remember selective in osmosis a while ago. At the beginning of this video, we talked about the concept of selection with what? Osmosis. So it means that useful substances, the ones you want, will be the ones that will be taken in, and the waste ones like your urea will be allowed to go all the way. And in tubular secretion, your antidiuretic hormone will now be key in making sure that the distal tubule is porous depending on the water level of the body. Okay, when the body needs more water, more ADH will be secreted to the distal tubule, because the body cannot afford to lose a large amount of urine because the body needs more water. Okay, the osmotic concentration of the body is high, so the body cannot afford to lose urine, so more ADH will be sent into the distal tubule, and that's going to make sure that the distal tubule is porous, leading to a very concentrated urine. So if there's something you want to write down now, write down the fact that more ADH means that you would have concentrated urine, and less ADH would mean that you would have dilute urine. Okay, so we move on from there. Now the next one is a table on the organisms and then the excretory structure and all of that. So you have amoeba, cell membrane, name of waste products here, ammonia, the flame cells for flatworm, ammonia again, nephridia is used, or nephridium, in earthworm, and they have urea there. You have malpighian tubules, and you have uric acid, you have humans, and you have urea. This table is very, very important. Pay attention to the concepts in this table. They will help you to do very well in your exams. Okay, so now we move to the next question. We are trying to be a bit fast because we still have some things to cover. Now, list 2 excretory products in plants, list 4 functions of the mammalian skeleton, and what is ecology. So we've come now to ecology. This is very important. And then we now brought this table to define some terms. We have species, we have population. You'll see the other ones on the other slide as we move on. So, but let's just quickly look at this together. This one here, you have the excretory products in plants are your oxygen and your carbon dioxide. Functions of the mammalian skeleton: supporting the body, protecting delicate organs, movement and locomotion, formation of the blood cells in the bone marrow. And ecology basically is a study of organisms in real and in their interactions with one another, with their environment. So study of living things in relation to their environment is basically your ecology. And then you have your species. Your species are organisms that can interbreed together. Please pay attention to this definition. They can interbreed, they can mate together and produce fertile offspring. All members of a same species living together in a particular area make up a population. For example, all the human beings in your class, okay, they make up a population of human beings. Okay, and a community basically is the different groups of different populations living together in a habitat. So that's why they would always ask you that a dead log of wood is a community. Why? Because in this dead log of wood you have ants, you have termites, you have mushroom. Different organisms will grow on that dead log of wood. So that is what makes it a community. Okay, so we move on to the next slide very, very quickly. The next slide says, okay, the other continuations now, the terms you have to define: community, biosphere, ecosystem, biotic, abiotic, ecological niche, pay attention to that, and habitat. Okay, and then here you have describe briefly how you can estimate the population of plant species using a quadrat. How do you use a quadrat to estimate the population of a plant species? And then here also you have state 2 reasons why ecologists use sampling techniques in population studies. So sampling techniques will be a key factor there as well. Okay, so we move on to the answers. So let's quickly see the answers to those questions. Now the answers here, we have that the part of the earth where life exists is your biosphere, of course. Ecosystem is the interaction, the community interacting with the environment, meaning that it's a make-up of the abiotic, of the biotic and the abiotic components. Basically, the biotic are the living components in the environment, and basically the biotic components are the producers, your consumers, and your decomposers, actually. Okay, so your producers, your consumers, and decomposers make up the biotic components, but the abiotic components are the non-living components, like your temperature, your rainfall, your humidity, and the rest of them. Okay, your ecological niche basically is the role or the functional role of an organism in its habitat. They ask this question a lot. Write down 5 roles played by a mango tree in its habitat. So that's like saying its ecological niche there. So you need to know that the function of the role or played by an organism in its habitat. This involves how it reproduces, what it feeds on, and where it lives basically. And of course, the habitat is the natural dwelling place of an organism. Okay, we have three types of habitats in biology. You have the arboreal habitats, the ones that live on trees. You have the aquatic habitat in water, and you have the terrestrial habitat on land. So how do you use a quadrat? You have it there. It was asked in your WAEC exam. I'm not going to repeat that, so please just go ahead to run through that. Now, state two reasons why ecologists use sampling technique in population studies. Number one, it saves time and labor, and number two, it is impractical to count every organism in a large area. So please pay attention to those facts there. This will really help in your exams. I hope you've subscribed. If you have not subscribed, please go ahead, click on the button to help this video to go as far as possible. The next question says, describe the characteristics features of the vegetation of the tropical rainforest with two examples of plants found there. So tropical rainforest, we are going there and we're going to see the examples of plants we have there. So this is still part of ecology. Give two local Baltic communities in Nigeria and associate each type with a particular state in Nigeria. And then we have the next one. Explain briefly the biological basis of preserving foods using each of the methods: salting, irradiation, and smoking. And then you also have here, what is a food chain? What is a food chain? So let's quickly see the answers in a bit. So for the answers, basically for the tropical rainforest, you have a very high amount of annual rainfall, over 200 cm. It has a dense vegetation with tall trees. The trees form the distinct layers. You have your stratification there in the forest. So the story, the underground layer, the canopy layers, and all the layers you have there. So you have your evergreen broad-leaved plants, and then you have your high humidity throughout the year. Examples of plants you would have in your tropical rainforest would be your oil palm and your mahogany. The other ones I just had to list down those two. And local Baltic communities in Nigeria would be your mangrove swamp forest, like what you have in Rivers State, and you have your Guinea savanna, like what you have in your Kwara State. The other ones, you have your Sudan savanna, you have your Sahel savanna, you have your mangrove swamp as well. So please pay attention to those and look out for the answers as they may come in your exam. Now, the process of preserving foods using the method salting. The salt helps to remove microorganisms, okay, by osmosis, like we talked about earlier, preventing their growth. Irradiation is when you expose the food to radiation, and that helps to prevent microorganisms and destroy, prevent spoilage and destroy microorganisms. And for smoking, you're using heat actually. The moisture helps to dry out the chemical and the moisture, inhibiting microbial growth. So pay attention to those few explanations. And a food chain basically is a sequence through which food energy passes from one organism to another. Okay, simple feeding relationship that shows the transfer of energy from organism A to organism B. That is a food chain. Okay, so we are making progress. The next question says, draw a typical grazing food chain and a detritus food chain involving 4 trophic levels, okay, in a terrestrial habitat. The next one says, explain the flow of energy in a food chain, like the one you will draw in the question here above. And then, how is the energy lost to the environment? Now, this one came in your WAEC exam. I had to bring it here again. State the first and the second laws of what? Of thermodynamics. I hope you still remember what you stated in your exam. Alright, so let's quickly see the answers. So here, a detritus food chain, it's a grazing food chain starts with grasses. So you can say your grass to your grass. They basically mentioned 4 trophic levels. A trophic level is a particular position of an organism in a food chain. So 4 trophic level means you have 1 grass. So you go to grasshopper, then you go to termites, and then what can prey on termites? Let's say snakes. Okay, yeah, you can see snakes there. And the detritus food chain is any food chain that starts with a decomposing matter. Okay, so you can say your dead leaves, and then dead leaves, you go to your termites, and then you can go to your small bird, and then you can now go to your snake. Okay, so that becomes a detritus. So the main thing to know here is that the grazing food chain starts with a grass, whereas a detritus food chain starts with a decomposing matter. Remember, there is no sunlight when you are drawing a food chain. This is the producer. The first member is always the producer, occupying tropic level 1. The second one, the grasshopper and the termites in this case, occupy tropic level 2. And the third one, the termites and the small bird here, occupy tropic level 3. And in this case now, snake was the one in the 4th tropic level for both of the food chains, so that occupies tropic level 4. I hope you are taking note of these things. They are very important and will come in your exams. So explain how the flow of energy is how it happens. So energy flow from the producers to the consumers. Like I mentioned, these producers now, the grass now will help to push the energy to the grasshopper and the termites and the snake. Okay, so that's what I have here. So how is energy lost? These are questions they like asking. It can be lost in respiration, movement, heat loss, and excretion. Okay, so that's very, very important. You need to know that. And then the first law of thermodynamics, basically, this law says that energy cannot be created nor destroyed, but can be transformed from one form to another. That's what you wrote in your exam, right? And then the second one says that during the transfer of this energy, efficiency is not equals to 100%, meaning that some energy is lost as heat in the process. Okay, so examples of plants and animals. Okay, two examples of plants and animals each that live in a fresh water pond. So basically, for the plants you have the water lily and the duckweed, and the animals you have the tilapia fish and the tadpole. Those are the examples you have there. Okay, so we move on to the next question. Let's try to see what we've got in the next question. The next question says, name 2 examples of plants and animals that live in fresh. Okay, we talked about this already. What is an estuarine habitat? Okay, state 3 zones of a marine habitat, stating one characteristic of each zone. So you have the 3 zones here and the characteristic feature. We are going to see that in a very short while. Now here you have the first diagram. Make a diagram 8 cm to 10 cm long of the reproductive system of a human female and label fully. Okay, so let's quickly see that in a short while. So here basically you have an estuary habitat is a body of water that's where the fresh water from the rivers mixes with the salt water from the sea. So basically it's two-sided. Fresh water comes and meets with the salt water. That's what we mean by an estuary. So check out the characteristics of a typical estuarine habitat in case you are asked in your exam. For your marine habitat, you have different zones. You have the littoral zone, that's the shallow water near the shore, and then you now have the neritic zone. In the neritic zone, you still have light. Light can still be accessed by your plankton. Plankton and lower microorganisms you have in water. You can be the ones that are photosynthetic are called phytoplankton, but the ones that are not photosynthetic that depend on the plant photosynthetic plankton are called zooplankton. But you now have the oceanic zone. Here it's very deep, where you have your big, your whale, your shark, all those very dangerous fishes. You have them in the oceanic zone of the marine habitat. So basically, this is a diagram showing the female reproductive system. Basically, you have, let me just use this to explain some basic things. Ovary, now that's where the eggs are produced. So the eggs are produced in the ovary. Fallopian tube, this is where fertilization takes place. Fertilization simply means the fusion of the male and female gametes to form a zygote. So the egg come out. When they come out, the sperm cell also come in from the vagina and they travel all the way, and somewhere on the fallopian tube they meet, and that is fertilization. When they meet, if it is fertilized, it comes down to the uterus. So the uterus is the womb. Okay, another name for the uterus is the womb, and that's where the baby is implanted. So that's basically the function of the uterus, for implantation and for development of the baby, okay, inside the body of the mother. And then the vagina basically is where the sperm cells enter into, the penis enters into the body of the female, basically depositing the sperm cells and also from the birth canal where the child is given birth to. So I think we've touched the basic parts of the female reproductive system. So let's move on to the next question. Alright, so moving on to the next question, we have here, name 4 functions of the human placenta. We also have the name 3 adaptations of the human sperm to its function. So you can see that these slides are not just careless questions. We touched as many questions, as many topics as possible, just to make sure that we help you and contribute to your becoming very, very confident as you prepare for your exam. So we ask here again, oviparity, ovoviviparity, and viviparity. You need to know how to define those basic concepts. And then here we ask, describe the digestion of a boiled fish in the gut of humans. So you have to also know how to talk about that. Let's quickly see the answers to these questions. Functions of the placenta: you have transfer of oxygen to the fetus, you have transfer of nutrients to the fetus, you have removal of fetal waste. Of course, when the baby in the womb excretes, it is through the placenta that the waste will be sent to the mother's body or the mother's bloodstream. And then production of hormones for pregnancy maintenance, especially progesterone. Okay, that's very, very important. And then the 3 adaptations of the human sperm cell to its function. It has a long tail because it has to travel long distances. I showed you earlier, from the vagina all the way to the what? To the fallopian tube where fertilization takes place. Numerous mitochondria. As it's traveling, it needs energy, as a matter of fact. So that's why it has numerous mitochondria in its middle piece. You can just browse the structure of the sperm cell to see these things for yourself. And then you now have the acrosome. The acrosome contains enzyme for penetrating the head. On the mouth and the head part of the sperm cell, you have that acrosome, and that's what helps to dissolve the jelly coat in the egg for the penetration, for fertilization to take place. Very interesting process. Now you come to oviparity. Oviparity is for animals that can lay eggs, and this is basically they lay the eggs outside the body of the mother, like what you have in your hen or your chicken farm. Your ovoviviparity, here the eggs develop inside, but before they give birth they just come out. Okay, they don't have a placenta actually. Okay, and you have that in sharks and also in some reptiles. And in viviparity, the young develop inside the uterus and receive the nourishment from the mother. And example is what you have in your humans. Now the next question is, describe the digestion of a boiled fish in the gut of humans. Basically, the fish protein is chewed, and because it's protein, digestion starts in the stomach. Remember, digestion of starch starts in the mouth, but for protein now it will start in the stomach. Okay, so here pepsin will help to digest the proteins into peptones. We talked about this earlier on. And in the duodenum, trypsin will further break down the protein into peptides. Okay, so we're going to see the balance or the remaining part of the question, the answers, in the next slide. Okay, so basically here, differentiate between filter feeders and fluid feeders. And then here you have complete the table on the arteries, the veins, the erythrocyte, xylem, and phlegm. Some of these words may appear new to you, but you should be able to write them down. That's why we are bringing this video, so you can just try your best to see how you can put these things together and help to prepare for your exam. Okay, and then what is transpiration? What is transpiration? You should know that as well. So let's quickly look at the answers to these questions. So we're talking about the digestion of the fish. Basically, erepsin converts peptides into amino acids, and basically the amino acids is absorbed in the bloodstream. The villi helps in the absorption of all the digested food substances. The villi has different parts. Okay, the villi has the capillary, and the blood capillaries help to absorb your glucose and your amino acid. But the lacteal, there's a part of the villi called the lacteal. The lacteal helps in the absorption of fatty acid and glycerol. Now differentiate between filter feeders and fluid feeders. Fluid feeders are organisms that strain the food particles from water. Okay, so they have like a mesh that helps to strain the food particle. But the fluid feeders obtain fluid by sucking, basically, like what you have in your mosquito. So the whale is a filter feeder, whereas your mosquito is a fluid feeder. The difference is that the filter feeders, they sieve the food particles from water, whereas the fluid feeders will suck the liquid food. Here you have the table of the blood vessels we talked about earlier on and some other parts. So you have your arteries carry blood away from the heart. Basically, most arteries carry oxygenated blood except the pulmonary artery. You should know that at this level. And then most veins also carry the oxygenated blood except the pulmonary vein. Veins carry blood towards the heart, okay? And then erythrocytes are your red blood cells. Of course, you have the leukocytes, which are your white blood cells, and you also have your blood platelets, which are otherwise known as your thrombocytes. And then the xylem helps to conduct water and mineral salts in plants, and your phlegm conducts manufactured food in what? In plants. So please take note of that. Write these things down. What is transpiration? Is a loss of water from the aerial part of the plant, especially the leaf, basically. Okay, so you have different types of transpiration, like your stomatal transpiration, your lenticular transpiration, and your corticular transpiration. Okay, so moving on to the next question. Name 2 types of transpiration. Talked about that already, and 4 factors that affect the rate of transpiration. We'll see that in a while. And then here we were asked to draw and label the mammalian heart and explain the pulmonary and systemic circulation. Let me just quickly talk about this. Pulmonary circulation is the interaction or the flow of blood between the heart and the lungs. Okay, when blood goes from the heart to the lungs and vice versa, from the lungs back to the heart. Okay, that is what? Pulmonary circulation. But systemic circulation, the second one, has to do with the flow of blood from the heart to the body cells. Okay, heart to the body cells and back from the body cells to the heart. That is systemic circulation. Okay, so take note of that. But we'll see the structure of the heart in a bit. Now, using a simple diagram, explain the nitrogen cycle. We're going to talk about that also in a short while. So let's see the answers as we move on very quickly. So we talked about the different types of transpiration. We're not going back there. Stomatal, corticular, and lenticular. The factors that affect the rate of transpiration: temperature, humidity, wind speed, and light intensity. These are all factors you should know. Now, this is a diagram of the heart. You should just know how to draw or sketch this diagram in case you are asked to sketch it. But this is the right atrium, this is the right ventricle, the left atrium, the left ventricle. Just study the parts of the heart, as we don't want to make this video very long. Okay, so study that and try to see how you can read, get some extra marks on that. Okay, so moving on to the next question now. We were asked to talk about the nitrogen cycle. Okay, let's see the next question before we go back to that. Now here it says, describe 3 processes involved in water and carbon cycles. So we're talking about the 3 cycles: nitrogen, carbon, and water cycles. These are called biogeochemical cycles, and they are very important for your preparation. So here you have this table of parasitism, mutualism, and commensalism. What are these things? These are symbiotic relationships. They are type of relationships, and let me just give you a little light on them. In parasitism, it's a one member gains, one member loses kind of relationship. So a parasite is preying on the host, right? And then in mutualism, the both parties are gaining. Remember earlier in this video I talked about the lichen. That lichen is a mutualistic relationship. And then commensalism, one member is gaining and the other member is neither being harmed nor gaining anything. So it's just being indifferent. So that's the difference between parasitism, mutualism, and commensalism. They are both types of what? Symbiotic relationships. So let's quickly see a few of those answers. So looking at the answers here, you have that the nitrogen cycle is a continuous movement of nitrogen between the atmosphere, the soil, and living organisms. So it involves, you must know these stages: number 1, nitrogen fixation, nitrification, assimilation, feeding, ammonification, and denitrification. These steps are very, very important. Okay, all those processes are the processes that lead to the form of nitrogen that is taken up by plants is NO3, and that is called nitrates. Okay, whereas the form of nitrogen in the atmosphere is N2, that's atmospheric nitrogen, has about 78%, but even this 78% is not needed by the plant. So the plant must always look for ways of converting from this atmospheric form to nitrates before it can use it. So it's already here. So go and read up this stage and see how you can understand it for yourself. If you have questions, you can put them in the comment section. We're always in the comment section to interact with you and to answer all the queries of your heart. Okay, so the processes involved in the water and carbon cycles are your evaporation, your condensation, and your precipitation. Okay, know that and prepare for your exam. The next one, for carbon cycle, you have photosynthesis, respiration, decomposition, and combustion. These are all processes. All of these processes release carbon into the environment apart from photosynthesis. Photosynthesis is the only process that takes away carbon from the environment. And for the water cycle, all the other processes, like, okay, there is also transpiration. Transpiration is actually from plants, when plants release water into the environment. Okay, so that's the part of the water cycle. All right, so moving on to the next question. I know the information is getting plenty and plenty, but don't be tired. That's what it takes to be a smart learner. Okay, so what is courtship behavior in animals? And moreover, we're also going to be bringing this to an end very soon. So list 3 examples of courtship behaviors, and then now we are asked to complete this table on the mosquito larva, the dodo plants, and the housefly and the toad, basically the adaptation for obtaining food. We also have this mimicry. Mimicry is a very interesting part of your behavior in animals. You're going to see that in a very short while. So let's see what we've got there. So here for this relationship, we have that tapeworm in the gut is parasitic. Yes, mistletoe and the cacao plant is parasitic. You have the lichen here, you have your flower and honeybee here. You have your remora and shark, and you have your cattle and your what? Your white egret. So these are all examples of the cattle is not necessarily being helped by the white egrets. Go and know what each of the members of this relationship do. Just try your best to study and read about that. Now, courtship behavior is a series of activities performed by animals before mating to attract and select mates. Okay, so the peacock spreading its feathers, proud as a peacock, so that's where it comes from. And then the croaking of the male frogs, and then making all that noise it does, basically just to attract the mate. And then the courtship dance in beds. These are all examples of what courtship behavior. And then here you have the different adaptations for obtaining food. For the mosquito larva, you have that. The dodo plants, you have that. The housefly and the toad, you also have that. Okay, so basically you need to pay attention to this as we move on. All right, so moving on to the next question. Give 2 reasons why termites are described as social insects. Why are termites described as social insects? And then complete this on the case system and the role played by termites. Basically, there are 5 members of the caste system in termites, so we are going to see that very, very quickly. Okay, so let's quickly look at that. So here you have that mimicry is the resemblance of one organism to another organism for protection or advantage. Basically, let me tell you about this. The mimicry, the organism that mimics the other one, maybe the main organism it has something like a distasteful, it is not very tasteful, maybe it tastes very bitter, or it has something that can sting the predators. So this other organism, I've learned from experience that if I try to mimic this other one that is harmful, my own predators will leave me alone. So that's the concept of mimicry. Okay, so give 2 reasons why termites are described as social insects. They live together in colonies. They exhibit division of labor among a case system, basically. Okay, and then you have this as their beautiful classification. These guys are very organized. There is this division of labor. Everybody knows what he's supposed to do per time. So the king fertilizes the queen. Wow. And then you have the queen laying all the eggs. The workers build the nest and gather the food. The soldiers defend the colony, and the alates, I love the alates, they're the wing reproductive. They establish new colonies. Okay, when the colony becomes too large to contain, the wing reproductive will travel on a nuptial flight. They will go very far and they will establish a new colony for themselves. So that's basically what you have there in the termitarium or in the termite case system. So please stay note of that and pay attention to all those beautiful things there. All right, so moving on to the next question. What is eutrophication? Very important. Eutrophication is very, very important to you. State 2 causes of eutrophication, and then briefly explain endangered species and threatened species. You need to explain that very briefly. And then what is conservation? State 2 reasons for conservation of natural resources. And then we have this table on the different functions of the parts of the human brain, cerebellum. You'll see the other ones as we move to the other set of the slides there. So let's quickly see our answers. Organisms that exhibit Territoriality, you have them here: dog, lion, lizard, where they protect their territories. You need to see a dog protecting his territory. That will be very funny. And then you have organisms exhibiting seasonal migrations. You have the swallow, you have the whale, and you have this wild beast. Now, organisms exhibiting display, you have the peacock, the turkey, and the bird of paradise. There are other ones, but these are just the ones we listed here for you. Eutrophication is the excessive enrichment. Pay attention to this. Excessive enrichment of water bodies with nutrients, leading to rapid growth of algae. Now, let me explain what happens here. In eutrophication, you have a farmland that is very close to a water body, and then you apply fertilizer, your NPK fertilizer on your farmland, and there was a rainfall, and very quickly the rain washed away all the fertilizer into the water body. And because of that high level of nitrate, remember we just stated that nitrates are needed for plant growth, right, in nitrogen cycle. So because of that high level of nitrate, what happens is that the growth of algae has been increased in the water bodies, and that will block sunlight from the lower plants. So the lower plants will die. There's going to be the bacteria will increase rapidly, using up all the oxygen, and then all the organisms in the water will eventually die. It's not a good thing. Eutrophication is a very bad thing for water bodies. So the causes basically are the runoff of fertilizers from farms and discharge of sewage. Yes, sewage can also be a factor. Remember, sewage contains a lot of urea, and urea is rich in nitrogen. So that can also be a very great source of that eutrophication. Now, explain endangered species and threatened species. Endangered species are those number of organisms that have become very low and are at the risk of extinction. Okay, so you no longer see them every time, and before you know, they will become extinct. And threatened species are also very close to becoming what? Endangered. So that's the difference between them. So what is conservation? Conservation is a wise management and protection of natural resources to prevent depletion. So two reasons for conservation: you have to prevent extinction of species and to ensure that resources are available for future generations. These are the two basic reasons for what? For conservation. So moving on to the other parts now. The parts of the human brain, you have the cerebrum that controls intelligence and memory and voluntary actions, the cerebellum that maintains balance and coordination, the optic lobe that controls vision, the olfactory lobe that controls the sense of smell, the medulla oblongata that controls involuntary activities like breathing and heartbeat, and then the hypothalamus that regulates body temperature and water balance, and the pituitary gland, of course, we talked about this earlier on, that secretes hormones and controls other endocrine glands. So this is the reflex arc. Now you need to know parts of this reflex arc. So you have the stimulus as this candle light, and the receptor cell on this hand takes the impulse, and then the sensory nerve, there you call it the sensory neuron or the afferent neuron. So take note of that. Sensory or afferent neuron would send the impulse to the central nervous system, which is the spinal cord, because it needs a very quick response. So the spinal cord will take the response, and then there is something called the relay neuron or the interneuron in the spinal cord. Okay, so the interneuron interprets and sends the impulse back to the motor neuron. So you call it the motor or the efferent neuron, and then the effector is always a muscle or a gland. So that's the sequence from the stimulus to the sensory or afferent neuron to the interneuron to the motor neuron to the effector and then to the response. So please write that down. They might ask you that. So the sensory neuron carries the impulse from the receptor to the central nervous system, whereas the motor neuron brings the response from the central nervous system to the effector. The effector carries out the response. So pause this video, write down those points, and you'll be fine at the end of the day. Okay, so moving on to the next question. We are almost getting to the end of this session. So we've explained this already with the reflex arc. So moving on to the next question, explain how a bitter taste is detected in humans, and then describe how the mammalian skin can regulate the body temperature in a hot room. And then you also have what is ecological succession, and describe the process of primary succession on a bay land. So basically we're going to have this explained to us. You also have here what is variation. So let's quickly see the answers to this in a very short while. So here you have that a sense organ is a specialized organ that is made up of receptor cells that detect stimulus and transmits impulses to the brain for interpretation. So explain how bitter taste is detected in humans. The bitter substance dissolved in saliva, the taste buds on the ground on the tongue are stimulated, the sensory nerve carry impulses to the brain, and the brain interprets the sensation as bitter. And then here you have how the mammalian skin can regulate the temperature of the body in a hot room. So you have the sweat gland producing more sweat. The concept here you need to pay attention is vasodilation, because vasodilation happens during a hot day, and in vasodilation the blood vessels close to the surface of the skin will dilate. Dilate means they will expand, and then the ones farther away will do what? Will constrict, and blood will flow to the top parts of the body, and that's what will lead to increased sweat, and your body will be cold. Okay, so please pay attention to that. And then what is ecological succession? Ecological succession is what? Is the gradual replacement of one community by another until a stable or a climax community is formed. So the primary, we have two types of succession. We have the primary succession and the secondary succession. Primary succession could occur on a bare land, a land that has not been cultivated before. So I listed out the points for you here, that this is colonized by lichens. There's what we call primary colonizers, the first set of organisms to be found in a particular uncultivated area are the primary colonizers. So now you have your lichen breaking down the rock to form the soil, and then the mosses would grow on the soil. You now have the herbs would appear, and small animals will be coming in that are supported by this growth of plants at each stage of this succession. So you now have your shrubs develop, your trees forming a climax community. So you need to pay attention to that at the end of the day. Okay, and then finally, what is variation? So variation is the differences that exist between individuals of the same species, right? So some can be tall, some can be short, some can be fat, some can be slim, some can be fair. Basically, these are all variations between individuals of the same species. So you have these terms on genetics, very important. Please go ahead to read genetics, read questions, learn how to carry out all those Mendelian cross in genetics, because they might come in your exams. Okay, if you have enough time, we are going to put up another video on the basics of genetics crossing, but just go and learn about the monohybrid cross and the dihybrid crosses, and all those things could come in your exam. They actually brought the monohybrid cross in the exam they wrote in 2023. So it's very important. If you can check the past questions, go ahead and look at that, and then it will really help you to do well in your exams. So these are the basic definitions as relates to genetics. Just pause the video, put them down, and then you'll be fine at the end of the day. It's been a very long one, a very long one with us, but I know you got so much from this video. As a matter of fact, this was a very good in-depth revision for you. It's going to really help you to prepare, and at least it will bring your confidence level to a particular point that as you get into the exam hall, you'll be able to write and do very well in your exams. Okay, so this is a disclaimer to tell you that these questions were carefully selected from the examination portals to help you do well in your exam. These questions are not the real questions you'll be seeing in your examination hall, but they serve like a revision guide to help you properly structure yourself and go into the exam. If you revise the questions very well and do all the things you're supposed to do properly, you will be able to come out of the exam hall smiling and happy with yourself that you did well. So thank you very much for your support so far. It has been massive, and I really appreciate how you guys have supported us in this channel. If you are yet to subscribe, kindly help us to click on the subscription button and like, share, and send these videos to as many persons as possible, so when next we make a new video, you will be the first to receive the notification.